There are approximately 400,000 species of plants known to science, in which there's a huge diversity of different structures, sizes, and shapes, say from the very smallest grasses to the very largest trees. However, within this diversity, they are united in the fact that the vast majority of them get their nutrients through the roots from the soil, and also synthesize their own sugars through photosynthesis using energy from the sun and of course carbon dioxide and water. In today's episode, we are going to explore a relatively small subsection of the plant world that can get some of their nutrients and energy in a different way. These of course are carnivorous plants. Plants that trap and digest insects or some other creatures for food. So in today's episode, we are looking at carnivorous plants and mainly looking at the different types of plants that we see, how their trapping mechanisms work, and a little bit on how they evolved to get here today. So let's jump straight into things and look at some of the basics with carnivorous plants. So carnivorous plants trap and digest mainly insects, although they do eat other things as well, after which they absorb their nutrients, which they can then use to grow and just survive, so maintaining their biomass. These nutrients are nutrients that all plants need, so nitrogen, phosphorus, and potassium containing compounds, as well as other trace elements. So these are just compounds and elements which all plants need for the manufacture of key components within them. Think of your proteins and DNA, but also for regulating key functions such as the water potential. So you need ions, for instance, to maintain water balance within the cells. Most plants, however, absorb almost entirely these compounds directly from the soil. So as you are well aware, all plants have pretty extensive root networks, high surface area with tissues adapted for absorbing these nutrients as well as water from the soil, which they then can uptake, transport throughout their tissues and use to manufacture key things such as those proteins I was talking about. And as with other plants, carnivorous plants derive the majority of their energy through photosynthesis. So photosynthesis is that process where we produce sugars using sunlight, carbon dioxide and water. They are also able to absorb some of the sugars and energy through the digestion of insects that they are then used to, they can use for energy. Just like how animals absorb their energy through the small intestine, what animals eat, those sugars, for instance, of fats can then be absorbed and used for energy. Humans obviously cannot photosynthesize. But this amount of energy absorbed is typically fairly small into relation to the energy synthesized by the plant through photosynthesis, as digestion is quite slow for cold-bodied plants. And if there's enough light for the plant, and there's water available, and there's the nutrients we spoke about, and you have access to carbon dioxide containing air, it's fairly easy for a plant to undergo photosynthesis. Thus the majority of the energy comes from this and not actually from the insects itself. So when you talk about carnivorous plants eating insects for food, we are not talking about them really getting much energy from those insects, but it's those key nutrients, micronutrients, that we're talking about them absorbing. So those NPK, for instance, when you talk about fertilizers for plants, nitrogen, phosphorus and potassium in compounds which are able to be absorbed. So for instance, nitrogen as a gas, it makes up the vast majority of our atmosphere, around 78%, but that nitrogen triple bond is very hard to break, requires a lot of energy to do so, and so plants can't really do that. At least most plants cannot convert nitrogen into a form that they can use. Some plants can, nitrogen fixing bacteria for instance, so you need to have it in a relatively absorbable form, in form of say ions or nitrate ions. And as you can expect, as carnivorous plants have evolved to obtain nutrients from other sources, or from sources other than soil, they are adapted to live in regions where the soil nutrient content is poor. So these regions are usually wet and boggy soils, but they are found on all continents across the world, other than Antarctica. It's a little bit cold there for plants in most cases. And in the UK, there are in fact multiple species of carnivorous plants native to here, and they live in wet boglands. So next time you're out in a bog or in a marsh, stuck after taking a wrong turn on a walk, have a look out for some carnivorous plants. Of course, many more have been imported because they can survive in greenhouses, so this is why you often see them when you go to botanical gardens. And how many species are there? Well, there are at least 800 species of carnivorous plants currently known to man. Of course, that value is likely higher because many more species are yet to be classified. And of these 800 or so species that we know about, they come in several different forms of trapping mechanism, which we will discuss the different types and the main types later on. And these plants are classified both by their physical structure and their evolutionary history. So while all of them are angiosperms, which are just the flowering plants, they are split across 6 different orders, meaning many are actually distantly related. And this is a classic example of convergent evolution. So carnivory wasn't inherited from one single ancestor, but was rather invented or evolved independently by different plant groups multiple times, and that number is thought to be at least 12 separate times that carnivorian plants has independently evolved. So if this is the case, what qualifies a plant to be carnivorous? Because many plants produce different chemicals, for instance, that are designed to kind of kill insects that eat them and insects that are harmful to them. So what makes a plant carnivorous rather than just a killer plant? So there are various traits that qualify, and if the plant meets this criteria, then they can be considered carnivorous. So firstly, the plant must be able to capture prey in traps. The plant must be able to kill that captured prey and then digest it. That prey, now broken up into key components, has to be able to be absorbed, and those nutrients can then must be used to grow and develop. So I guess that last criteria of using those nutrients to grow and develop means the plant's not just doing it for fun, but it's using those nutrients to actually survive and grow. Let's have a look at the different types of carnivorous plants. And there are 4 main types I want to talk about in this episode. And these, when I say types, I'm talking about the different trapping mechanisms here, because not all of the plants within the same trapping mechanism are very closely related. So I'm going to start with pitcher plants. I'm sure most people will have heard of pitcher plants because they are fairly distinct and there are many different types. So there are 2 main families of pitcher plants. I'm not going to pronounce the names because I'm just not very good at pronouncing the names of these. I don't want to get it wrong. And yeah, if you did look at the names of these plants on paper, some of them you might understand why I don't want to try and pronounce them. They're just I did try and listen to how they're meant to be said. Some of them even after that, not going to be able to pronounce them. So I'm not really going to try. So yeah, there are 2 main families. One family has 1 genus with over 100 species, and they are found all across Asia. And another family has 3 genera, and they live in the Americas. And the pitcher trap has evolved separately many times, so that's the convergent evolutionary. They've evolved independently the same trap, which kind of makes sense if we look at the 100 species across different families and different genera. And there are different forms of the pitcher plant, so some are very small and some are very large, but you can imagine they all look fairly similar, with the leaves all folded around to make a jug-shaped structure that's kind of tall and narrow, with a large bulge at the base where the insect falls, and an opening at the top for it to enter. So it's characterized by this internal chamber, which is the pitfall trap, made of these specialized leaves which curl round to form this vase jug structure that traps the insects. And it's a passive trap, so there's no movement involved by the plant. The plant just sits there and essentially just waits for insects to kind of come around it and then fall in. So the gist is they have to lure prey to the edges, so the very top of the chamber, where they can fall in. The pitcher is full of digestive enzymes, which provide a good environment for the enzymes to work, so correct pH, for instance, so digestion can occur efficiently. So what prey? Well, most of the prey they consume are insects and spiders, but some of these plants are actually quite large and have been known to catch even smaller mammals, such as mice, which is kind of crazy. When you think of carnivorous plants, you are thinking of insects and flies flying around, kind of getting trapped in them. But for something like a mouse to get digested, even if it's a small mouse, it's quite crazy. I feel like a mouse is just a different level. So how do they work? Well, first, because they are passive, they can't just go out and hunt, obviously. So they have to draw their prey in to them. The prey has to come to them. So insects are drawn to the entrance of the cavity right at the top with several different lures, such as the visual pigments, colourful pigments, some of which are visible in the UV spectrum that insects can see. So the colourful red pigments in the visible spectrum, as well as colourful pigments in the UV spectrum that insects can see. And as well as this colour, they also lure them with kind of nectars and food and sweet smelling smells. So how does the capture work? Well, the outer rim and the walls of the pitcher are pretty slippery due to the presence of these waxy scales. And these waxy scales kind of wrap around the ends of the feet of the insect, and so they really reduce the ability for the insect to stick onto the walls. And that, as well as the moisture on the walls, leads to them slipping into the pitcher. As well as this, there may be additional adaptations to prevent their escape. So once they're already fallen in, to prevent them from being able to escape again. And these can be, for instance, downward facing hairs. So downward facing, they can fall into the pitcher very easily, but going against those hairs is very challenging. And also brittle crystal-like structures on the inside of the walls that can break upon any pressure from the insect. So insect trying to crawl out and it steps on this kind of brittle crystal and falls back into the pitcher. And the inevitable outcome of this is the insect is trapped in this pitcher, which is full of this digestive fluid with enzymes and acid, and eventually the insect will drown and the digestive process will begin. So the insect is going to be basically dissolved and dismantled by various means. So we've got bacteria, mutualistic bacteria in the pit that secrete digestive enzymes, or enzymes secreted by the plant itself. And the fluid within the pitcher is not just any fluid, it's kind of a bit viscous and it's quite acidic, and it reduces the ability for the insect to escape, but also it kills it faster, and the acidity provides a good environment for the enzymes to work. So that you can think the pH is actually quite low, it's quite acidic, so pH 2 to 3, which provides an optimum environment for those enzymes to function. Kind of like in the same way in the human stomach we have hydrochloric acid, which is the same acid found in the pitchers, and those kind of two-pronged effect of the acid. Well, it kills any bacteria, or most of the bacteria that come into it, but also that acidity provides an optimum environment for certain enzymes, specifically ones that digest protein, to function at an optimal conditions. And some pitcher plants also have these mutualistic insects, are able to eat the captured prey themselves. Somehow these insects are able to survive themselves. But yeah, they, in certain plants, digest these captured prey and kind of act as the digestion for the plant. So they digest it and excrete this nutrient containing excrement, which the plant, well, it's already processed, so the plant can then just absorb it. So it kind of helps the plant in that way. And just like in our digestive system, the products of digestion are these nutrients in the form of amino acids from proteins that have been digested, sugars, some micronutrients such as phosphorus and nitrogen containing substances. And to absorb these nutrients, the epidermis of the leaf and the inside are specialized to have membranes, membrane transport proteins for these smaller molecules such as our glucose, our sodium ions, our hydrogen ions, ammonium ions, potassium ions, all these essential ions. So these transport proteins are specific to these ions and transport them across the membrane from the bowl, the jug, the pitcher, into the leaf itself, which they can then be transported around. As well as this, to maybe be a bit less specific with the absorption, because these transport proteins are very specific for certain compounds, the plant can absorb by a process called endocytosis, which just means that the nutrients or whatever is in the pitcher can be encapsulated into these small membrane-bound spheres, which we call vesicles. So the membrane of the outer layer, the epidermis, kind of folds over itself, folds over and encapsulates the products of the digestive process. And these vesicles can then remain inside the cells. And the benefit of this is it's quite non-selective. And by the way, this process of endocytosis, where the nutrients are encapsulated in these vesicles, is the reverse of the process called exocytosis, which is used to expel the digestive enzymes used to digest these compounds in the first place. And like other carnivorous plants, pitcher plants grow in areas with poor soil nutrients, and it's important for them to get their micronutrients such as nitrogen and phosphorus from insects, and their energy on the other hand is got mostly from photosynthesis, where the plant manufactures their sugars using carbon dioxide from the air and water with energy from the sun. So although plants still use the sugars they digest, the majority comes from photosynthesis. And upon researching this episode, I found a paper on the digestive system of carnivorous plants just generally, and it said this quote, which I kind of think is quite good at summing up how carnivorous plants operate. So the carnivorous plant leaf is an all-in-one organ, kind of analogous to the animal digestive tract. And this sums up things pretty nicely, because in the case of the pitcher plant, the leaf is responsible for intaking the insects, which is the mouth, as well as the secretion of enzymes and hydrolysis of the compounds by those enzymes, which happens in the stomach, as well as absorption of nutrients, which occurs in the small intestine. So it's a kind of all-in-one organ that does everything as an animal's digestive system. Although I would say it happens slower because they do not produce heat, so the enzymes work faster at warmer temperatures, but these enzymes are just working at whatever temperature is in the ambient environment. So that is pitcher plants. Now let's have a look at snapping traps. So what types of snapping traps are there? Well, there are two main well, they're two different types, the Venus flytrap and water wheel trap. And these two plants are quite closely related, and the snapping mechanism has only evolved once in history. So the Venus flytrap is native to wetlands of North and South Carolina in the US, and we'll focus on the Venus flytrap for this episode because it's closely related to the water wheel plant. So what does the trap look like? Well, I think most people know what a Venus flytrap is. I think it's the classic example when people think about carnivorous plants, just think of the Venus flytrap. But the trap has two kind of lobes attached to this midrib, which is the central spine structure of the leaf. And there are trigger hairs on the lobes. So on the inside of those lobes, they're kind of reddish colour. They have three, in the case of the Venus flytrap, 3 trigger hairs on each lobe. And once triggered, these lobes kind of snap shut, trapping the prey inside. And what is this prey? Well, because these traps are smaller, we're kind of restricted to insects and spiders, so no mammals in this case, not quite going to fit inside the traps, although if it's a very, very small mouse, then I don't know, it depends how small they get. But yeah, the traps aren't massive, so we're not going to be trapping mammals really in most cases. Again, just like the pictures, although they are active traps, although they had to put in energy for this snapping process, they cannot move, so they cannot just go out and hunt, so they have to attract their prey to them. So the inside of the leaves have these colourful red pigments. You'll notice it when you see these carnivorous plants, they have this classic kind of red pigment inside. And these pigments, they essentially there to attract prey. And each lobe has 3 hairs. So upon an insect or spider touching these hairs, the trap will snap shut. But I think the trapping process is actually quite smart. So to avoid triggering and shutting of the trap without any prey inside, the trap actually requires 2 hair stimulations within about 20 seconds of each other, or 1 hair stimulated twice within a very short interval for the trap to snap shut. So this saves the plant from unnecessarily shutting its traps without having anything inside. So for instance, if a falling raindrop comes and hits one of those hairs, it doesn't just close, because the plant does not want to waste energy on this mechanism if it's not going to get anything from it. And the trap itself is pretty fast to shut, so it only takes about 1/10 of a second to close. It's a really rapid process. But if you think about insects, especially flies, they are so quick, they can change direction quickly, they can accelerate very quickly. This trap has to be quick, otherwise it's not going to have a high success rate in trapping. As well as this requiring more than 1 stimulations in a short period to shut the trap, to make sure there's prey inside, in order to start the release of the digestive fluids, an additional 5 stimuli of the hairs is required to ensure that the plant has actually caught a live animal and not just, say, a piece of debris that's kind of happened to fall and blow into the trap. And to make sure that the trap has caught a reasonably sized insect rather than the tiniest of ants, the trap has these kind of cage-like finger protrusions on the ends of the hairs. So when the trap shuts, these hairs kind of mesh around each other and leave these bars. And if the insect is small enough, then they can escape, and that saves the insect from spending energy on digesting such a small prey, which it's not going to get much value from. But larger insects are not going to be able to escape through these bars, and they're going to continue stimulating these hairs, which will push the trap further shut and start the digestive process. So how does this trap close? Because it's kind of cool. In humans, we have a nervous system which coordinates our muscles to move, but plants, very different. They do not have muscles, even though they're moving. In this case, for the carnivorous plant of the Venus flytrap, there are no muscles. So how does it work? How can the plant shut within 1/10 of a second its carnivorous leaf. And so yeah, in this section we'll have a quick look at how the Venus flytrap is able to move. So how does the trap close? Well, the trigger hairs act as a specialized touch sensor, so when they are bent by prey, they are instantly able to convert this mechanical pressure into an electrical signal. Very similar to how, well, for instance, in our skin we have these touch sensors that when you press, the mechanical pressure is able to deform the membrane and open these channels that allow ions to pass through and cause this electrical signal. So in the same way, we have this physical bending. It opens these gated ion channels in the hair base. So calcium and these other positively charged ions rush across the cell membranes. So the sudden shift in ion concentration across the membrane causes depolarization. So if we have a movement of positive charges across the membrane, that's going to change the charge across the membrane. This electrical shift is identical to the formation of an action potential in the nerve cells. So the result is we have this electrical signal in the form of action potential. It fires with a voltage of around 150 to 200 millivolts and travels across the trapped tissue at speeds of around 10 meters per second, so it's pretty rapid. And the way the trap has this memory-like feature where it requires 2 stimulations within a certain time period for the trap to shut, is kind of works through this electrical summation. So with a single touch of the hair creates what's known as a sub-threshold charge, so not enough ions pass through to create this action potential, but these ions kind of still remain across the membrane. So if there's another stimulation of the hair in a given time period, it will cause more ions to move across the membrane. And once those do, the charge from the first stimulation is added to the charge of the second stimulation is enough for a full action potential. So it's called electrical summation of the charges sum. And because these closing mechanism is driven by electricity, scientists can basically apply a voltage across the trap and shut it purely using voltage. So now we've got this electrical charge kind of propagating up the lobes of the leaf after stimulation. How does that cause movement? And there are 2 main theories here. First is known as the acid growth theory, which is kind of where we have this action potential triggering a rapid pump of hydrogen ions into the outer cell walls of the lobe, which drops the pH and loosens the cell walls and forces water inside by osmosis. The outer layer of the leaf will then swell as more water comes into it and expand, while the inner layer stays the same. So you can imagine the outer wall gets longer, whereas the inner wall stays the same, and that kind of forces the trap to buckle inwards on itself and shut. The other theory is kind of the other way round. It's called the Turger loss theory. So the electrical signal travelling up the leaf, same, causes ions in the inside of the lobe to then kind of dump ions out into the surrounding space. Water immediately flows out of these inner cells via osmosis again, causing them to shrink and get smaller. So as they get smaller and get shorter, the inside of the lobes will then, well, they'll get shorter, whereas the outside stays the same, and it has the same effect, whereas the trap will kind of collapse shut. So these are kind of 2 different ways of getting the same result. So now the trap is shut, how can we digest it? Well, it kind of works the same way as the picture. They cannot escape and they continue to stimulate the hairs. The trap kind of seals itself shut like a stomach, and we'll get this release of digestive enzymes. The hydrolytic enzymes, they break down the insect and they'll then kind of absorb up any products of digestion. And this process usually takes around 10 days, but it would vary depending on the size of the insect as well as the temperature. And after this digestive process has occurred, the trap will reopen for ease. Okay, so that's Venus flytraps. Now let's have a look at our third kind of trap, and that is the flypaper trap. So flypaper traps have evolved independently at least 5 times, and there are 2 main genera here. So we've got the sundews and the butterworts. So these genera contain several hundred species in total, so just over a couple of hundred species. And they're characterized by their use of the sticky mucilage, which is this thick, gooey polysaccharide kind of solution which is used to trap insects. So flypaper kind of flies stick to this mucilage, and they're stuck and they can't get out, and then they can be digested and nutrients used. So the traps are covered with this mucilage secreting network of glands, which may be short, so close to the leaf, or relatively long, and so kind of separated from the leaf, or kind of higher up, with the mucilage kind of secreted as droplets further up. So I'd encourage you to have a look at some of the photos if you get the chance on Google. Just have a look, and look at the different kinds of these traps. So the mucilage being sugar, it's naturally already a bit attractive to insects because they want to use that as food, so they are attracted to that when they stick. And so after the prey is trapped, some species actually can fold the leaf over the prey and kind of help stick it further and to prevent it from being washed away by any rain. Some as well are able to fold over to form this kind of digestive pit. It speeds up the digestive process with these digestive fluids kind of held in this pit. And as the same as the other ones, once the prey has been trapped, digestive enzymes are released to break down this prey, and nutrients are absorbed through these little holes in the leaf surface. These holes also increase water loss through evaporation, so these plants typically live in more humid environments. So let's start with the sundews. So the sundews can attract their prey because they appear quite shiny and wet. So insects that are thirsty and hungry for nectar that they produce, sweet nectar, are attracted. And these are the ones with the long tentacle-like structures that secrete the mucilage at the end, and so the insects become stuck when they land on the leaf. And then we have the butterworts. They also appear slightly wet to attract the insects with the sticky mucilage, but the difference here is their glands are closer to the surface, not projected up. But the process is fairly similar, so digestive enzymes break down and nutrients are absorbed through the cuticular holes in the leaf surface. Now, let's look at our final type of trap, which I didn't really know about before researching this episode, and to be fair, I think it could be the coolest trap, and that is the bladder trap. So there's only one genus of bladder traps with over 200 species within this genus, and they're typically located either in fresh waters or in wet soils. So we've got terrestrial species that live on land, and they have relatively small traps to trap pretty small prey, such as the protists. And then we also have aquatic species that tend to have larger bladders to catch larger prey, so insects and sometimes also some fishes. So how does the trap work? Well, the bladder pumps ions out of its interior by active transport using energy, and when ions are pumped out, it causes water to move out by osmosis and generates some partial vacuum inside the bladder. So you can imagine a sealed box almost, that while water leaves, no air can get in, so we generate a vacuum. And this bladder has some entrance that's covered with a hinged door, so when an aquatic insect triggers the hairs, the door hinges open and the insect is drawn into the bladder by the vacuum. So got an area of very low pressure inside of the bladder and relatively higher pressure outside. So we have this pressure gradient, and in order to equalize that pressure gradient, when the hinge opens, water floods in to that vacuum along with any insect around the entrance. So once the prey is sucked into the trap, the door closes again, and the body of the bladder is thin but pretty rigid, and that's required to stop caving in under the vacuum. And in order to ensure there's a seal around the bladder for the door, the cells around the door produce this kind of mucilage, sticky substance that kind of seals acts as a sealant, but also is full of sugars to attract any insects around the entrance of the bladder. And around the door there are these antenna structures which kind of act like tripwires that guide prey into the door, and when the tripwire is activated, it causes the door to open. The point of activity, the active point, is not at the point of triggering, because here it's a purely mechanical process. So energy is used to set the trap by pumping ions out. That uses energy through active transport, but then the trap holds that potential energy in the form of pressure gradient. So the trap is held in this stable state, but when the trap is triggered by the triggering of those hairs, that causes the door to open, and the system returns to a lower energy equilibrium state. So think of it like releasing a spring held stretched mechanically. So the trigger hairs are just levers. Pushing against them breaks the seal and opens the door, pulling water inside the insect. It's not active at the time of the trap opening, but it's active at the time of setting the trap. The whole process is complete pretty quickly because it's a vacuum, strong vacuum, a high pressure gradient, water sucked in very quickly, and the whole process completes in around 1/100th of a second, and the door resumes its closed position. So once the prey is inside, again we have to see the same thing. We get enzyme secretions, it dissolves, breaks up into components. Often there are these mutualistic bacteria, just like in the pitcher plants, which can help digest some material that the plant cannot digest, and the bacteria then excrete some products which the plant can absorb. So those are the types of carnivorous plants I wanted to cover in this episode. In this final section, I'm going to just cover some questions that I was thinking about when researching this episode. So firstly, why don't more plants have some carnivory? Wouldn't it be useful to have multiple methods of obtaining nutrients? So other than just using nutrients from the soil, uptaking through the roots, wouldn't it be useful to have multiple ways of obtaining them? So my understanding of this is that when conditions are good, meaning the soil has adequate quantities of nutrients, the small benefit of having extra nutrient gaining capacity is not worth the effort of having traps. Thus, this adaptation is not advantageous, and so it's not selected for. So we don't see this evolution for plants that already have adequate nutrients uptake through the soil. As well as this, it may be disadvantageous because the trapping of pollinators may not be great for long-term success. Another question I had is, when do these carnivory traits evolve in plants? So we know that they have evolved at least 11 or 12 different times across 6 different orders and 20 genera. So there is some form of convergent evolution, so separately evolving similar features. And molecular phylogenetics has dated the evolution of the oldest known lineage of plant carnivores to around 95 million years ago, but there is evidence that older carnivorous plants may have lived as long ago as 120 million years ago. But it seems that most lineages originated between 8 and 72 million years ago. And looking at evolutionary history of organisms typically relies fairly heavily on the fossil record, so preservation of tissues and bones and parts of plants or animals throughout time as fossils. But with carnivorous plants, the record is pretty sparse. So firstly, carnivorous plants do not readily fossilise, as they're made of softer tissues compared to those with harder structures, such as stuff found in wood. It can make it quite challenging to get an accurate picture of how they have evolved over time. So yeah, that is everything I wanted to cover in this episode of the Curiosity Monkey podcast. I hope you enjoyed this episode. I certainly learnt a decent amount from researching carnivorous plants. I especially like the bladder trap, and maybe I'll get some carnivorous plants of my own. It's also interesting to find that they're kind of located everywhere across the world. I kind of thought of them as more a tropical plant that lives in more tropical conditions, but it's interesting to see that there are many species native to Britain as well. So yeah, I'll definitely be looking out for some next time I'm stuck in a marsh. But yeah, that's everything for this episode. Hope you enjoyed it. If you have any episode suggestions or feedback, please contact us. So thank you for listening, and see you in the next episode.